Welcome back to Scientia Academiae. Today, we're diving into some fascinating questions of Biology Paper 2 June 2016 session. So, let's get started. Question 16 is about industrial manufacture of single-cell protein and carries 10 marks. First up, in part A, let's explore the industrial manufacture of single-cell protein, a valuable source of nutrition. The process begins by sterilizing all equipment and raw materials to ensure a contaminant-free environment. Once that's done, it's crucial to monitor and adjust the pH to create the ideal conditions for our microorganisms. Next, we introduce sterilized materials into a fermenter. Here, maintaining an optimal temperature is key, often achieved with a cooling jacket. We then add our chosen organism, be it a bacterium, fungus, or algae, along with a nutrient-rich growth medium containing glucose and ammonium salts. To keep everything well mixed and oxygenated, we introduce bubbles into the fermenter. Once the biomass has grown sufficiently, we separate it from the liquid medium using filtration or centrifugation. The final product, mycoprotein, can serve as a meat substitute or livestock feed. Now, let's look at the challenges of using viruses in biotechnology. While viruses have great potential, their small size makes them tricky to handle. Additionally, they can only replicate within living cells, complicating large-scale applications. Many viruses are pathogenic, raising safety concerns, especially in therapeutic uses. Lastly, isolating them from living tissues can be quite labor-intensive. These hurdles definitely limit the effective use of viruses in biotechnological advancements. Question 17 is about understanding drugs and carries 10 marks. Now, let's define what a drug is. A drug is a specific substance or chemical administered externally, whether through oral intake or injection. Its primary role is to modify the body's chemical reactions, leading to various physiological changes. These interactions can yield therapeutic benefits or adverse effects, emphasizing the need for understanding how drugs function in biological systems. Now, onto a critical health issue, smoking tobacco. Tobacco smoke contains several toxic components. For instance, nicotine is highly addictive, enhancing dopamine release and leading to dependence. Carbon monoxide reduces the blood's oxygen-carrying capacity by binding to hemoglobin, while tar can cause severe respiratory issues, lung cancer, and cardiovascular diseases. The health implications are significant, highlighting the dangers of smoking and the impact on overall well-being. So now let's talk about, why is smoking increasingly viewed as socially unacceptable? Well, health concerns about passive smoking, particularly for vulnerable populations like children and pregnant women, play a huge role. The lingering odor and the high cost of tobacco further diminish its appeal, making smoking seem outdated and irresponsible. Moving on to question 18, which carries 10 marks. With reference to the inheritance of blood groups, explain what is meant by dominance and codominance. First, dominance. This means only one allele needs to be present in a heterozygote for the trait to show. For example, in blood types, the alleles Ia and Ib are dominant over the recessive I, leading to types A, B, and O. Then we have codominance, where both alleles are expressed equally. In a person with genotype IAIB, both A and B antigens are present, resulting in the AB blood type. Finally, let's discuss chromosomes. Gametes contain half the number of chromosomes, 23, making them haploid, while body cells, or somatic cells, have a full set of 46, or diploid. This difference is crucial for understanding reproduction, as fertilization restores the diploid number. That's all for today's episode. If you found this information useful, don't forget to like, subscribe, and hit that notification bell for more insights from Scientia Academiae. Thanks for tuning in, and see you next time.